My name is Smita Kurup, and I'm a senior scientist, and I'm also head of bioimaging, and I work at Rothamsted Research, which is a research institute that works primarily on agricultural research, and it's based near St. Albans. And my research is got a couple of different focus. I do a lot of fundamental research on seed biology, where we're interested in looking at seed yield and how to increase yield. And the reason for this research is really the translational aspect, which is for agriculture. So in terms of global food security, as we're well aware, this is a topic that's discussed quite frequently. The idea is we have a maximum of land that is under cultivation at the moment. And what the problem is going to be with this increasing population in the world and people living longer, we're going to need more food. And so the idea is by 2050, if you're going to need enormous amounts of extra food, it's going to be impossible to actually cultivate more land because most of the arable land that is cultivated is being cultivated. So the only other two other options are we increase yield. So we want to get more yield per hectare of land, and also we want to reduce pathogen and diseases because that is another way of increasing your yield per hectare. So with these ideas as the sort of driving force for our research, we look at all of different aspects of doing research to increase yield, whether they come from a fundamental angle or they come from a very crop-based angle. And we want to use a variety of different approaches to get to our end point, which would be to increase not only yield, and sometimes when we talk about yield, it's not just increasing bulk yield, it could also be the quality of the product. So for example, even if you're talking about wheat grain, rather than having, if you want more fiber, you want to increase the amount of soluble fiber, which is digestible fiber, rather than just fiber per se. So sometimes we call this nutraceuticals. So the idea is you increase the good in any kind of food tissue. So you eat the same amount, but you get more of the good stuff, rather than having to eat twice the amount of spinach, which sort of people don't want to do. So the idea is for the same amount of spinach, for example, if you can get more mineral nutrition from it, you're still going to have a healthy diet. So these are all the different approaches we use in Rothamsted Research to sort of tackle food security. I'd like to tell you a little bit about some of recent research that I've started, which is on waxes in plants. And what happened was I worked both on fundamental model plants, as the Arabidopsis, and also brassicas. And in brassicas, when we were looking at the plants, what we found is a lot of plants, including brassicas, have what we call waxes. So the leaf of a plant, if you go into your garden, you will see there are some plants that look quite green in color and some that have a bluish haze on them. What plants have is on top of the cuticle or the top layer, they have a layer of laminar wax. Now this is normal, every plant has it, and the idea of the laminar wax is it helps protect the plant, helps with water stress, etc. Some plants, in addition to this flat layer or laminar wax, have a crystalline layer of wax which we call epicuticular wax. This epicuticular wax is what gives it that bluish color because it reflects a lot of the blue light. And what we got around to thinking was, why does the plant need this extra layer of wax crystals? Does it have any real benefit to it? Because we thought, well, if it's reflecting a lot of light back, that is light that the plant could absorb. And as you know, plants need light for photosynthesis. So my colleague Fred and I, when we started discussing this issue, we thought, well, this is interesting. Is it possible that we could somehow reduce this cuticular wax layer? And what would be the effect on the plant? Our idea was that there would be increased light into the plant, and therefore a higher level of photosynthesis, higher photosynthesis should give you higher yield. So we did this as a pilot experiment. So we were working on brassicas, and we took a brassica plant, and what we did was, you can use good imaging techniques to really look at this wax crystalline layer, and it really looks like a sort of skyscraper. When you see a scene of New York skyline, it really looks like that. These are these crystals of wax which is on top of the surface of a leaf. And what we did was mechanically we removed this layer of wax, and then we use a machine which measures photosynthetic rate. And when we put that on at different light levels, we saw that the rate of photosynthesis increased by up to 15%. And now this percentage was even more obvious at lower light levels, which is interesting if you imagine we live in Northern Europe. Northern Europe, as you well know, our summers are very cloudy. We don't get very high levels of sunlight sometimes. So the fact that even when light levels are quite low in nature, the idea is the plant could absorb more light and convert that into photosynthetic carbon. And what will happen is eventually if you have saturating light, I think the level of photosynthesis would be equal whether you had your waxes or not. And that was what we discovered. We found that it was amazing that at very low levels of light also, the photosynthesis could go up anywhere between 5 and 15%. And this photosynthesis, we hope, will now get converted into, in brassica, we are looking to convert it into oil in the seed, which would mean we could increase the oil in brassica or canola seed by up to 15%, which would be 15% sounds low, but when you think that a brassica plant, Brassica napus, makes about 50% oil in the seed, increasing that to 65–70% would be a big economic boost to farmers who grow them. We were wondering whether there would be any disadvantages to removing the wax, because you sort of think if a plant has evolved an idea of having the wax, perhaps it has some reason to be there. Is it protection against the environment? Is it protection against pathogens? And interestingly, what we found is there are actually pigments within the epidermis of the cell or in the wax layer, the laminar wax layer, which are the ones that really absorb harmful UV radiation. So this crystalline wax layer does not seem to play a role in protecting the plant. The second thing was we thought, will it affect the water stress quality of the plant? And again, we found it's the laminar wax which makes the difference, not the epicuticular wax. And then the third thing we came around to thinking was, pathogens. Maybe somehow they play a role in stopping pathogen attack. Now, this is an area that we've only had a very preliminary look-see, and we've looked at some publications that are out there. And this is something we want to further look into. But our idea is actually, rather than being a disadvantage, it could actually be an advantage because predators of pathogens are able to move around much faster when the leaf doesn't have any wax. So it's more of a smooth surface. So the predators can get around much faster and therefore attack the nasty insects. So we think actually, even in terms of pathogen resistance, this is going to turn out to be quite a good thing. So the other obvious reason also is in gas exchange. So we think there will be actually more gas exchange. So again, there will be more carbon available for carbon partitioning into, again, oil. So as far as we can tell at the moment, most of the things are advantages. We haven't come across any disadvantages, but as in science, we need to prove all of these before we can say we are there. In terms of getting this research out to farmers, at the moment what we want to do is refine our knowledge. So we want to really find out how we can modulate the waxes. So we want to identify genes or loci that will make these levels of wax go down without any other detrimental effects. So, and there are different approaches we can do. We can use very targeted gene approaches, or we can do forward genetic screens that will bring us to the same endpoint. And both of these are what we are considering. In terms of the UK and EU market, we're very aware that non-GM approaches are generally much more acceptable for the general population. So we also have an idea that we can use non-GM approaches such as tilling, where tilling is a natural mutant population that is available in brassica and other crops. And what we would do is find natural mutants in our genes of interest, and these mutants would be the ones that we would test. And the other advantage we would have with this is also you would have mutants in more than one gene. So what the advantage could be that if you cross these mutants with each other, we might get an added beneficial effect. So this is where we'd go. And the idea with the tilling mutants is once you know that there is a gene which if we mutate, you will get a beneficial effect, this is when breeders can use this information. And what they do is use marker-selected breeding. So they make markers to your gene of interest that you want to have mutated or increased, whichever way. And they go and look at their breeding population and screen for particular lines that have the effect they want. And then they'll have a breeding line which reciprocates the sort of gene of interest that we want. And that's how a breeder would get it out to the market. And if we get to the point where a breeder has it, you can imagine in a few years, 3 to 5 years, they could have a breeding line that would be able to grow. The UK farmers would be able to grow. What I'm hoping is at the moment, for a new generation of scientists, what we want to be able to do is accelerate the pace of research. Technology is moving at an incredible pace, and at the moment, moving from research in the lab to something in the field takes close to 10 years. And that pace isn't good enough if we really want to feed the world in the next 20, 30 years. So what we would like is a situation where scientists in a wet laboratory situation are more aware of companies, are more aware of breeders and stakeholders, and we actually work together rather than a scientist doing research in a lab and then going and talking to a breeder or a stakeholder. What we need to do is have communication right at the start so we know exactly what a breeder is looking for. So we incorporate that in our research. And that's what we've been doing with this project with the waxes. We have spoken to a number of companies and breeders and discussed our ideas with them, which helps because they give us ideas, for example, about the pathogen and to look for things. And they realize how changing the wax layer might be useful or not, because breeders traditionally have never looked at the epicuticular wax layer. They look at the laminar wax and they have never really concentrated on the epicuticular wax. So they're quite interested that there might be a little thing that we could do to modulate, which wouldn't affect the plant per se, but would give us a beneficial effect in terms of yield.